Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub, and today's point of discussion is how to derive an acceptable intake for new N-Nitrosamine. So this presentation is prepared based on to the guidance provided by US FDA and EMA as per as nitrosamine is concerned. So as a part of risk assessment, you are going to understand whether there is a possibility of formation of nitrosamine or not. And in case if your primary assessment says that, okay, there is a chance of getting nitrosamine into your product, then you need to test the product for the content of nitrosamine. And as a part of your analytical exercise, you understand that, whether there is a presence of nitrosamine or not. The first question you must begin with is N-nitrosamine found into your product? And if the answer to this question is, of course, based on to your practical experiment, the answer is no, there is no presence of nitrosamine into your product, and you must be happy about it that you need not to include the nitrosamine into the specification. You need not to have the nitrosamine as a part of your specification. But in case if there is a content of nitrosamine found into your product, then you need to understand whether there is a TD50 value available for this particular nitrosamine. The TD50 value stands for the, the concentration of the substance at which the 50 percentage of the population will develop a tumor. So if you begin a study with, let us say, 70 species, so understand the concentration and the time at which the 30 species will develop a tumor, and that particular concentration, the exposure is called as the TD50. And this TD50 value is provided by the CPDB database, the carcinogenic potency database. It is available with the LASA now, and it is also called as the LASA standard or the gold standard. So get the CPDB value if it is available, the robust TD50 value if it is available from CPDB database, you are lucky to have that. So if the value is available, calculate the acceptable intake by linear extrapolation from TD50. And how the extrapolation has to be done to understand what is the concentration or the dose which will result in a tumor in only one species out of 1 lakh studied species throughout the entire lifespan. And that is the meaning of linear extrapolation from the TD50 value. So once you have that, you can define the limit. But in case if the TD50 value is not available, in case if the robust TD50 value is not available, then here is the approach. Can N-nitrosamine be controlled at default limit of 18 nanogram per day, which is defined by the EMA as a threshold for nitrosamine group of compounds, or 26.5 nanogram per day as defined by US FDA? Now this is very conservative approach. In case if you do not have any kind of toxicological data, if you do not have TD50 value, then can you able to control the content of nitrosamine at this particular level, either 18 nanogram per day dose or 26.5 nanogram per dose? If the answer to this question is yes, then you can happily control the nitrosamine at those particular levels. But if the answer is no, if your process is not able to control the nitrosamine at 18 nanogram per day, which is proposed by EMA, or 26.5 nanogram per day, which is proposed by US FDA, then what you can do? You can certainly derive the acceptable intake based on to the structure activity relationship or the read across approach. Now, this approach is well explained by US FDA and EMA, by both the regulators. And if you want to have some information about that, just look into the US FDA and EMA nitrosamine guidelines, and you will find that there are number of examples where the SAR or read across approach is used. For example, in the given table, you will find the limit for NDMA as 96 nanogram per day. I mean, the acceptable intake for NDMA is 96 nanogram per day. And if you look at the acceptable intake given for NMBA, yeah, let me take the pointers over here. So if you look at the acceptable intake given for NMBA, it is also 96 nanogram per day. So it is read across from the NDMA. And same is the case for NMPA, NIPEA, and NDPA. So all these molecules does not have the TD50 values or the robust TD50 values. Only two molecules, NDMA and NDEA, have the robust TD50 values. So based on to structural activity relationship or read across approach, US FDA has derived the limit for the following nitrosamine. So in case if you can also use this approach and try to identify, you know, the acceptable intake. And if this acceptable intake gets accepted by regulators based on your scientific explanation, then certainly you can happily go with the controlling those nitrosamine by derived acceptable intake based on to SAR or read across approach. But what if this SAR approach is not accepted by regulators? What if it is not, it is not accepted by the regulators? Then you can think about the in vitro AIMS test. Now, AIMS test is the in vitro test which is proposed by ICH M7 guideline. And according to this guideline, AIMS test is going to understand whether the molecule is going to be a mutagenic or a non-mutagenic. But if you look at the nitrosamine, these regulators, most of the times, they do not accept the AIMS test data. Whether your AIMS test result comes negative, positive, it doesn't make any sense, and you are forced to conduct the rodent toxicity study. Now, this rodent toxicity study is going to determine TD50 values. We talked about TD50 values 3 to 4 points back in the presentation. Let us now understand, in case if you want to perform the rodent toxicity, how you can do that, how to conduct a rodent toxicity. So now this is again proposed by the EMA guideline. You need to conduct the rodent toxicity at multiple doses, like low, medium, and the high. Let us say these are the examples. That dose 1 contain 2 milligram exposure, dose 2 contain 4 milligram exposure, and dose 3 contain 6 milligram exposure. Now this is just for your understanding and reference. Then at least the three groups has to be considered for this exposure study. Let us say that these are the three different groups, the group 1, group 2, and group 3. So according to this guideline, minimum 50 animals per dose and per sex should be a part of each and every group. So the group 1 contain 50 male rodents and 50 female rodents. Group 2 contain 50 male rodents and 50 female rodents. And similarly, group 3 also contain 50 male and 50 female rodent. So each group will get exposed by three different doses. So group 1 will get exposed for 2 milligram dose. Group 2 will get exposed for 4 milligram dose, and group 3 will get exposed for 6 milligram dose. So out of this exposure study, you will understand whether the drug substance or the compound under investigation is positive or negative. Let us talk about the negative part first. Negative means what? There is no evidence of formation of tumor is observed. That is the negative test. And in that situation, you can classify this compound as a class 5, means no mutagenic characteristics observed. In that situation, you can control this particular impurity substance according to either ICH-Q3A or Q3B. But in case if the okay, I think yes. But in case if the result is found to be positive, where there is a formation of tumor is observed, that means your substance looks to be a possible human mutagenic. So in that situation, you need to calculate the TD50 value. And based on to TD50 value, we can calculate the acceptable intake by linear extrapolation with the point of understanding what is the required dose which will result a tumor into one species if 1 lakh species gets exposed throughout its entire lifespan. So this is the way you can decide the limit for, or the acceptable intake for the newly identified N-nitrosamine. Thank you so much.